Hello, this is lecture 3 of the course Demystifying the Brain. In the last lecture we talked about brain structure, we talked about the relationship between brain structure and intelligence. We began with the study of Einstein's brain and we found that the study was inconclusive and the results were like not very interesting, not very significant. So, then we looked at other aspects of brain structure, particularly wiring diagrams, wiring patterns, and we found that wiring patterns are important because that can the wiring patterns can optimize the delays involved in transmission of signals from one part of the brain to another. So, we invoked this principle called save wire principle, and with that we were also able to account some of the gross features of brain's anatomy, right, through evolution, starting from simple creatures like hydra or to more complex brains like human brain. But if you really want to understand the question of brain's intelligence and brain function, you cannot go very far by just studying brain structural properties. You need to get into signaling, you need to get into the neurons function. So, that is what this lecture is about. So, in this lecture we will talk about neurons and neural activity or neuron function. Now, traditionally it has been thought that brain's function is or brain's information processing functions are basically implemented by neurons, these particular cells called neurons, but work done over the last couple of decades has shown that there is a second class of cells called glial cells, which also are involved deeply in brain's information processing activities. So, we will talk about these two kinds of cells, neurons and glia. Now, neuron is a special kind of cell, it is electrically active cell. So, it belongs to a class of cells called excitable cells or excitable tissues. So, they are relatively active and they also have this interesting wiring structures, wiry structures which project out of the cell body, right, these arboreal projections. And the number of neurons in human brain is about 100 billion, although the number has been revised. There is a new estimate which says adult brain has about 86 billion neurons. In terms of size, the cell body of a neuron, which is called soma, can range anywhere between 100 microns in case of a motor neuron of the spinal cord, or a small neuron like granule cells, it has only a 4 micron thick soma. But when we talk about size of a neuron, we should also, if you consider the entire arbor, then neurons can be very big. For example, a neuron sitting in your spinal cord, right in the lumbar area of the spinal cord somewhere here, can send a projection all the way up to your toe, which means the single cell is like as long as a couple of feet, right. So, neuron size can vary a lot. So, now let us get some intuitive understanding of what is special about a neuron shape. So, like here in this picture you are looking at a bunch of cells, you can see some bacteria like bacillus and paramecium, you can see the cardiac cells, you see those connections between cardiac cells, and the cardiac cells talk to nearest neighbors, they have what are called the shoulder to shoulder contacts by which they communicate with nearby neighbors. Then there is skeletal muscle, right, then there is red blood cells, there is donut shaped red blood cells and bone marrow cells and all that. So, typically the shape of these cells is nothing very interesting. I mean, the typical cell is like a fluid filled shapeless blob, but just look at some pictures of neurons, right. Neurons, one thing that strikingly dissimilar or distinct about a neuron is these wiry structures that stick out of the soma. In fact, in this picture it is very hard to find the soma, right. Soma is like a little dot somewhere in the middle of this in a complex tree, right. And then so these neurons, this picture show a variety of different kinds of neurons. There is this pyramidal cell on the top left, then you have a small gelatinosa cell, and then there is this on the top right you see this complex cell with very rich arbors. It is called a Purkinje cell. It is located in the cerebral structure towards the back of your brain, and an average neuron has about 10,000 connections, whereas a Purkinje cell has about 1 or 2 lakh connections, that is about 100 to 200,000 connections. So, that is what is special about a neuron, and here you see a typical neuron, actually it is a pyramidal neuron, right. A neuron has all these wires, but these wires are typically grouped into 2 sections. So, there are this shorter section, the short set of wires which are called the dendrites, you see in the upper part of the figure, and then typically you have a long wire called the axon, right, which towards the end it branches out into many branches. These are called the axon collaterals, right, and at the end of the collateral you have an axon terminal. So, between these two you have the cell body. So, you can see that unlike in other cells, the cell body is only a small part of a bigger structure called the neuron, right, and cell body is called a soma in case of a neuron, right. And so, on the axonal side you also have some neurons have this kind of a myelin sheath provided by certain other cells. They are a class of glial cells. Because of myelin sheath, the conduction speeds on the axon are faster, and the conduction is more reliable because the wave shape is more reliable, and we will talk more about this later on in this course. So, the neuron actually takes inputs from other neurons on the dendritic side, on the upper side, and processes and combines all these inputs that it receives from various neurons in the soma, and then at the soma it generates new signals which are propagated along the axon, and these signals run down the axon to the end of the axon, to the axon terminal. And at the synapse, right, which you see at the bottom of the figure, right, you see sort of a contact between a presynaptic neuron and a postsynaptic neuron at the synapse, and this is where the signal jumps from presynaptic neuron to the postsynaptic neuron to another neuron, and then this cycle continues. So, let us take a closer look at the synapse. So, at the synapse, in this picture you see one structure coming from the top, and this is actually a presynaptic terminal, the tip of an axon of a certain neuron, and it makes contact with the dendrite of another neuron, and you see that the point of the contact, which is actually the synapse, right, there is a very the presynaptic side has a kind of a swollen structure which is called the bouton, which is French for button, and on the postsynaptic side the dendrite is actually, although it is like a cylinder, you know, it is a tube, right, it becomes a flat structure, like a flat elevated structure, and the tip of the bouton is also flattened out, it is a flat surface. So, these two flat surfaces now meet at the point of synapse, right, making a coming into sharp apposition. So, that you have a lot of surface area at the point of contact, and that is necessary because if you remember from our first lecture, the signal that goes from the presynaptic side to the postsynaptic side is a chemical signal. So, the presynaptic side releases a chemical which is called neurotransmitter, and we will talk more about this later on in the course. So, this chemical diffuses through this narrow gap of about 20 nanometers, right, across what is called the synaptic cleft to the dendritic side, and then it acts on the dendritic side and then produces other electrical responses on the dendritic side. So, you need this kind of a special geometric arrangement at the synapse at the point of synapse, right, to enable this kind of signaling to occur in the synapse. Now, so that is something about the structural aspects of neuron. Now, let us come to the functional aspects, right. So, neurons, like I said already, that they are electrically active cells. Now, any cell has a certain potential, electrical potential inside it with respect to outside the cellular environment. So, if you take the outside as ground potential, inside in a neuron, in a typical neuron, it is about minus 70 millivolts. This number can vary depending upon the neuron type you are talking about, can be anywhere between minus 80 or minus 60 and depending upon the cell type, but the minus 70 millivolts is a reasonable typical figure. And this figure is not always constant. So, the neuron can remain in two states. There is a resting state, which is what it normally in, and in the resting state the voltage of the neuron inside the neuron is about minus 70 millivolts. This is called the membrane potential because this is a potential that exists across the membrane of the neuron. Okay, so, between the inside and outside, so what separates inside with the outside is the membrane of the neuron. Now, so resting condition, this membrane potential is constant, but when the neuron gets excited due to certain factors, we will talk more about that later in the course, right. Then this what was constant, this voltage shows very rapid fluctuations, as you can see in the figure on the right, right. The graph on the top that you can see consisting of these sharp spikes are actually these sharp voltage fluctuations, and these are nothing but the action potentials which we discussed in the very first lecture. So, that is when you say the neuron is active or the neuron is firing. So, the neuron can stay in two states, the resting state where the membrane potential is constant, the active state or excited state where the membrane potential shows as sharp spikes called the action potentials. Now, these electrical signals that are generated by the neuron travel along the wires of the neuron, along the axons and dendrites, and that is the signals that neuron communicates. Now, if you look at the complete picture of the neuron, so you think of the neuron soma as a black box, some kind of a black box, and dendrites carry inputs from other neurons. So, those are the input lines into the black box, and then all the signals are combined by the neuron, and new signal is generated by the neuron at the cell body, and this new signal that is generated travels down the axon to the axon collaterals and goes to the other neurons. So, you see that in this process there are 4 signaling components in a neuron. So, on the entry point of the neuron in the dendritic tree, right, all the signals propagate down the dendritic tree. So, that is the first component of signaling, that is signal processing along dendrites. Then all the signals come to the neuron and then they get added up, and an action potential or a train of action potentials is generated in the cell body. That is the second component, action potential generation in the cell body. Then these signals which are generated at the end of the soma or at the meeting point of the soma and the axon, which is called the axon hillock, these travel down the axon, and that is the third component, signal propagation along the axon. And finally, when the signal gets to the synapse, to the presynaptic terminal, right, it is converted into a chemical signal, right, which crosses the synapse. So, this signaling across synapse is the fourth component. Okay, so, that is broadly the signaling components in a neuron. Now, let us kind of try to learn that important lesson out of what we have just discussed. right. What is special about the brain as an organ, because this was the question that has been repeating right through all the previous lectures, right. What is so special about the brain? The question might sound somewhat naive, because you can just say that brain is designed to be special and brain is designed to be intelligent, but it is not as simple as that, because brain, like any other organ, is a bunch of cells, it is a mass of cells. Now any cell in the body has the standard infrastructure, like it has a nucleus, it has all these organelles like Golgi bodies and endoplasmic reticulum, and then it has a cell membrane and lipid bilayer, and there is nucleolus and all these things. So whether it is a neuron or a cardiac cell or a skin cell, they all have the same structures, right, internally. So what is special about the brain? So the answer to that, because you don't say pancreas is intelligent or heart is intelligent, you say brain is intelligent, right. What is the reason? Well, the simple answer to that is the neurons. There is something special about the neurons, and that is what makes the brain special. But then what is so special about neurons? Well, there are only two main distinguishing features of a neuron compared to other cells. They have these arboreal structures, right, they have these wires sticking out of them, and then they can exhibit rapid electrical signaling. So we have seen that the sharp action potentials, right, which are the sharp electrical spikes, are each spike is only about 1 millisecond wide. So these signals are very rapid. So no other cell in the body can display both these properties. No other cell in the body has such rich arboreal branching structures, right. Secondly, no other cell in the body can exhibit such rapid electrical signaling. Even cardiac cells have electrical signaling. I mean, that is why heart is an electromechanical pump, but those signals are much slower. For example, a cardiac cell fires an action potential of a cardiac cell is about half a second. That's very slow compared to a neuron, where the action potentials are like a millisecond. So basically, what is special about brain is it is a complex network, right. It is a network consisting of 100 billion units, and each of these units, the neurons, is connected to 1,000, 10,000 other connections and other neurons. And so this network is a dynamical network. It is sending these signals rapidly at the rate of sometimes hundreds of hertz, right. And also these connections are not fixed. It is not a fixed network. This is a labile network. It is a variable network. For example, the connections in your brain before the lecture quite different from the connections in your brain after the lecture. So at the fastest time scales, the connections among neurons can vary at the scale of like order of 10 seconds. That's very fast. Okay, so you are looking at a very complex network and a very labile variable network, and no other organ in the body can be described in these terms. So brain is a complex and dynamic network structure, and no other organ in the body has a similar network structure, because even if you take an organ like heart, which is capable of very complex dynamics, right, and very complex beat patterns. In the heart, the network is very simple, because each cell only talks to its nearest neighbors. So that is very simple, and it is fixed, and there is no change in the network. And whereas in the brain, a neuron can send projections to very far remote parts of the brain and talk to it, and this connectivity keeps varying internally. Although the gross structure of the brain looks about the same, it does not change like rapidly on the scale of days or weeks, right. Microstructure is changing constantly, right. So that is what makes brain very special. In this respect, you can compare the brain with the entire mobile network of the world. I mean, we have the planet has about 7.5 billion population, right. Among them, about 5 billion people are supposed to have mobile connections, and let us assume that each person has on the mobile like about few hundred contacts, right. I mean, that network is nothing compared to the network that you have right here inside your cranium. Okay, so in a nutshell, that is what is special about the brain, and it is really an amazing organ for these reasons. Now, like I said, there is a second class of cells called the glial cells, which also participate in the brain's information processing, right, in addition to neurons, right, and that is becoming more and more clear only over the last couple of decades. Glial cells have always been thought they are about 90% of the brain cells, but actually that means they are about 10 to 50 times more numerous than neurons. But more recent estimates have downsized these numbers. Looks like now people are thinking that for every one neuron there is a one glial cell. So the ratios are like more 1 to 1 and not 1:10. But glial cells do not carry nerve impulses, they are not electrically very active, but they show other signals, right, by which they seem to be responding to signals coming from outside, and also using these other signals they can talk to neurons. So there are glial cells found both in the central nervous system and the peripheral nervous system. Central nervous system, I think we have discussed this in the very first lecture, is the part of nervous system that is located, right, safely housed within this bony vault of your cranium, that is the skull, and the vertebral column. So that is like brain inside the skull and the spinal cord inside the vertebral column. Both these structures together is called the central nervous system. and then there is a peripheral nervous system consists of all the wires that go out of this bony vault and innervate the body and spread all over the body and innervate send signals all over to the body, that is called the peripheral nervous system or PNS. In the CNS there are three kinds of glial cells. There is the astrocytes, there are the oligodendrocytes and microglia. Now astrocytes like transport nutrients to neurons from blood vessels. So for example, I mean any cell in the body needs energy and that energy comes from the bloodstream in the form of glucose and oxygen. The thing is, a typical cell like for example a skin cell or a muscle cell, skeletal muscle cell, right, when it is active it spends energy, and so when it wants energy it sends out certain hunger signals which act on the nearby blood vessels, and the vessels dilate and then release oxygen and glucose, which is picked up by the tissue. But brain is different, I mean so neurons are different because they do not have some of the molecular machinery to process the energy that comes from the blood vessels. So they depend upon this kind of glial cells, the astrocytes. So astrocytes act as intermediaries between the blood vessels and the neurons. So you have astrocyte holding on to a blood vessel on one side and making contact with the neuron on the other side, and so it picks up the energy in the form of glucose from blood vessels. The glucose enters astrocyte where it gets converted into lactate. The lactate is supplied to the neuron where it is picked up by the neuron and then converted to ATP, and this ATP is used for driving the activities of the neuron. Now then there are oligodendrocytes. These are the cells which wrap around the axons of neurons and provide the myelin sheath so as to speed up conduction. I think we talked about some of these aspects in the last class, because we said that the myelinated axon fibers are faster than unmyelinated ones, and this myelin sheath actually comes from the oligodendrocytes in the CNS. Then there are microglia. These are like scavenger cells in the brain, and they clear the debris of dead neural tissue. So in the peripheral nervous system you have the satellite cells which act like provides physical support to the neurons, and the Schwann cells which also provide a myelin sheath to neurons, and Schwann cells are like equivalent to oligodendrocytes in the peripheral nervous system. So here is a simple cartoon picture of how a Schwann cell wraps around the axon over and over, right, providing a thick myelin sheath and thereby increasing conduction velocity. So there is this disorder called demyelinating disorder called multiple sclerosis, where patients do not have myelin sheath, right. This attacks 1 in 700 people, and because the sheath increases conduction velocity, loss of sheath reduces conduction velocity, and therefore these people have trouble with movement, with their perception and cognitive functions and so on and so forth. Okay, to summarize the relationship between neurons and glia, I think this picture is a simple cartoon picture that shows how the neurons are distributed or located with respect to the glial cells. So you can see that astrocyte, that green cell over there which makes contact with the blood vessel on one side and makes a contact with neuron on the other side, right, and shuttling energy molecules from bloodstream to the neuron. And then there is this purple cell which wraps around the axons of neurons, right, that is oligodendrocyte, right. This sheath is a myelin sheath, and that gives you increased conduction velocity on the axon. Okay, so the neurons and astrocytes talk to each other. So for a long time, like I said, people thought that glial cells do some kind of a secondary or insignificant functions. In fact, the word glia itself comes from Latin, which means glue. So people thought glial cells are just like glue whose purpose is to keep the neurons in place, but actually people found that they are very active cells. They participate in neural signaling. For example, it has been now known that astrocytes respond to neural signals. So neurons can talk to each other by exchanging chemicals, but interestingly it has been found that astrocytes can also respond to those chemicals. They have receptors for these chemicals, and then they can process these signals, and they can also release chemicals which can act on neurons. So they kind of get into the loop of neural communication cycles. So astrocytes also regulate signaling across neurons. So we have, so let us say 2 neurons, neuron A, neuron B, talking to each other across synapse. An astrocyte can send its end foot, right, or what is called the perisynaptic process, to wrap around the synapse and eavesdrop on what neuron A is telling neuron B. Okay, so and also it has been found that synapses form better in culture in presence of astrocytes. So astrocytes are very deeply involved in both the development and function of neurons and neural networks. So now the current thinking is in the field is that neurons and astrocytes form a tandem, work in tandem to perform brain's computations. Now, neurons are not the solitary heroes, right, that are doing all the brain's computations. Neurons are working along with astrocytes, right, to perform brain's computations. Okay, so we have talked a little bit about the brain's cellular constituents. Now let us talk about neural signaling. So like I said already, the neurons are electrically active cells, and these signals are propagate along the fibers of the neurons as electrical waves. Now, here there is a little difference in the manner in which these waves propagate on the dendritic side as opposed to axonal side. So, propagation over dendrites is usually lossy, that is you lose and the signals lose energy as they run along the dendrites, whereas propagation over axons is non lossy. So, let us look at that. So, here is a cartoon picture which shows a dendrite as a pipe, and then if you have a voltage wave, so starting at the leftmost tip of this pipe, as it propagates downward it loses amplitude, loses steam and loses energy, therefore, loses amplitude, and also it widens in time. Okay, that is why it is lossy, and this kind of a propagation happens on the dendritic side. Okay, so, now let us see what happens on the in the cell body proper. So, to understand that, right, let us imagine that you are injecting some current into the cell body. So, imagine that so forget about the arbors for a moment and imagine cell body is like a sphere, like a little ball, and then you poke it with a micro electrode, right, and inject some current into it, and let us say the current is the current pulse. So, the I of t here is a pulse. Now, what people have found is on something very special about a neuron is that when you give a current pulse, right, that little current pulse produces a small voltage response in the membrane potential. So, you are injecting current, you are injecting charge into the cell, so that increases the voltage, right. So, when your current pulse is small, that produces a small upward deflection of the voltage, as you can see in this graph, whereas now you keep on increasing your current pulse amplitude, right. As your amplitude increases, the upward deviation of the membrane potential also increases progressively up to a point. But beyond a point, when the current pulse exceeds a certain threshold value, you see that the voltage also suddenly shows this large upward swing, right, and that is the action potential. Okay, so therefore, this kind of response is called an all or none response, because it is like as the current is increased until a point there is not much response or very little response in the neuron, but beyond a point when the current crosses a threshold, right, suddenly it shows a huge explosive response, right, therefore, it is called an all or none response. And so, what we call action potential is actually a kind of an all or none response. So, this all or none response is actually produced at the meeting point of the soma and the axon. Okay, that is what is called the axon hillock, as you can see in this figure. So, once the action potential is generated at this meeting point in the axon hillock, it then propagates along the axon, as you can see in the cartoon picture at the bottom of this slide, right, and this action potential propagates along the axon, and in the top of the slide you can see this little kind of a pipe along which the action potential is propagating. So, if you measure the voltage at various points along the axon, right, at every point the action potential or the voltage wave has a fixed amplitude and width in time, right. So, therefore, as the action potential propagates along the axon, its shape is intact, there is no loss of amplitude and there is no spreading in time. But what happens on the dendritic side is totally different, and as it propagates on the dendritic side, it loses amplitude and spreads in time, whereas on the axonal side it is intact, right, it does not lose amplitude or there is no spreading in time. We will see why this happens later on and what kind of machinery that the axon has and why does it happen, all that we will see in the next segment of this lecture. So, here is simple animation which shows how the action potential propagates, right, along the axon. Okay, so, now we have come to the end of the axon at the meeting point of one cell and the other cell and at this synapse, right. So, if you remember from our very first lecture, we said that the signal that goes from the presynaptic side to the postsynaptic side is a chemical signal. Okay, so, this signaling step, which consists of transmission of a signal from a presynaptic neuron to the postsynaptic neuron, is called neurotransmission, and it is a chemical step. Okay, so, in this step basically what happens is you have the presynaptic side of a neuron, that is called the presynaptic terminal, and then the postsynaptic side of a neuron called the postsynaptic terminal. These two come together at the synapse. Now, on the presynaptic side, right, you have an action potential which marches down the axon and arrives at the presynaptic terminal. So, at that point some chemistry takes place, and we will discuss that later on in the second segment of this lecture. Some chemistry takes place and then the chemical is released by the presynaptic terminal, and this chemical arrives at the postsynaptic terminal by diffusing across the narrow gap of the synaptic cleft, and the chemical acts on the postsynaptic terminal and produces a voltage response, right, and this voltage response is called the postsynaptic potential for obvious reasons, which is called PSP. So, the electrical signal on the presynaptic side, the action potential, right, comes to the synapse, it gets converted to a chemical signal, and the chemical signal is again converted to electrical signal on the postsynaptic side, where it becomes another voltage wave which is called a postsynaptic potential. Now, these postsynaptic potentials are of two kinds, right. There are what are called excitatory postsynaptic potentials, right, where the postsynaptic membrane becomes more positive than what it normally is, that is it becomes grows higher than minus 70 millivolts, or what is called it depolarizes. And there is the IPSP or the inhibitory postsynaptic potential, where the postsynaptic cell temporarily hyperpolarizes, that means its potential goes below the normal value of minus 70. So, let us see how that looks. So, you have the two cases here. So, in the case shown in the top, the action potential comes in and which produces a PSP which is positive, right, above the baseline value of minus 70 millivolts. It is called the EPSP or excitatory postsynaptic potential. In the figure below, you have the action potential coming in, which is converted into again a chemical step and then the response on the postsynaptic side, where the PSP this time is negative, therefore, it is called the IPSP or inhibitory postsynaptic potential. Now, the PSPs, both EPSPs and IPSPs are graded potentials. That means, unlike the action potentials which have a fixed size and shape, the EPSPs come in all sizes, shapes and sizes. They can be positive or negative, they can be small or big, as you can see in some of these cartoon diagrams on this slide. Okay, now, when do you have an EPSP and when do you have an IPSP? So, that depends upon the nature of the synapse. So, there are two kinds of synapses. There are these excitatory synapses, where when you get an action potential on the input side, right, you get a EPSP on the output side or on the postsynaptic side, and there are these inhibitory synapses where you get an IPSP on the postsynaptic side. So, please remember that the signal on the presynaptic side is always the same, the action potential is always positive, it is an upward deviation from the baseline voltage. But on the postsynaptic side things are different, right, the voltage the PSP can be positive or negative, or it can be EPSP or an IPSP. So, in case of excitatory synapse you have an EPSP, and in case of inhibitory synapse you have an IPSP. So, now if you want to summarize this, right, we said neuron is like an input output system. So, the neuron soma is like a black box, receives input from dendrites, right, in the form of PSPs which are obtained at the which are generated at the level of synapses, and all these PSPs propagate downwards towards the neuron, and at the level of neuron, right, you have action potential just generated, and then which propagates down the axon to the axon collaterals, and at the synapse it jumps to the other neuron. So, you see that of these 4 signaling components, 3 components are electrical components. For example, signal propagation along dendrites is completely electrical. Then the summation of all these signals in the soma is also electrical. Then signal propagation along the axon, right, which is in form of action potentials, is also electrical. Only the 4th step, right, where the signal jumps across synapse is chemical. I just briefly mentioned before the neurons summate, right, different inputs coming from different neurons, right. For example, in this picture you see that large yellow neuron, it receives input from the red neuron and which happens to be an excitatory neuron. So, the synapse between the red neuron and the yellow neuron is an excitatory synapse. So, it is indicated by the plus sign over there, and synapse between the blue neuron, which is inhibitory neuron, and the yellow neuron is an inhibitory synapse, right, which is indicated by the negative sign over there. So, this summation that we are talking about in the soma is of 2 kinds. There is spatial summation, where it adds up signals coming from across many dendrites. So, how does that work? Let us look at these simple schematics. So, you have this big red region which is the soma of a given neuron, and it receives input from many other cells. So, you see that the red input lines are like excitatory inputs, and the blue input lines are inhibitory inputs. So, in this case 2 of the excitatory inputs are active and they are injecting currents and increasing the voltage of the soma. So, therefore, on the whole the soma membrane potential is higher, and probably this neuron will become excited and send action potentials. But suppose consider a case when the inhibitory synapses are also active. So, in this picture you see that 2 excitatory synapses are active and also 2 inhibitory synapses are also active. So, 2 reds and 2 blues, they all act and the blues counteract the reds, and therefore, on the whole there is not much change in the voltage of the neuron. So, the neuron may not fire, right. Look at this situation where there are 3 dendrites which are excitatory that are active and only 2 dendrites which are inhibitory, they are also active, right. So, on the whole you have 3 against 2, and so the membrane potential of the soma, right, maybe it sufficiently increases to become excited, and then it produces action potentials, and these propagate down the axon. Okay, so that is the kind of thing that is called the spatial summation. Now there is a different kind of summation that also occurs, where summation occurs over time. Okay, inputs add up over time, right, and at the same spatial location. How does that work? So in this picture you see a neuron, actually you can see the dendrite of a neuron, right, vertical line, and there is the axon of another neuron, the parallel horizontal line, which makes a contact with the dendrite of this neuron. Now the axon is carrying multiple action potentials, the whole train of action potentials are running along the axon, and all these action potentials arrive at the synapse and produce a PSP on the postsynaptic side. Now we have seen that the action potential is a very sharp spike, but the PSPs are much longer, right, running over like in maybe hundreds of milliseconds. So when the action potential hits, first action potential hits the dendrite, it produces a long PSP, and by the time that PSP dies off completely, the dendrite is hit by the second action potential. So therefore the PSP that is produced by second action potential now builds up on top of the previous PSP, and then the dendrite is hit by the third action potential. So by the time the second PSP has died off, right, the third action potential has hit the synapse, and therefore the PSP that is produced by it rides on top of the previous two PSPs. So because of this, right, the three PSPs add up to produce a huge wave, a very large ePSP. Okay, so this kind of a summation which happens at the same synapse, but because by integration or summation through time is called temporal summation. Okay, so to wrap up, right, what happens in a neuron is you have the cell body and it has multiple input lines, right. In this schematic you can see there is only one ePSP coming in, there is a topmost line, and then so because what you are seeing on the left side is are the dendrites. So there are multiple input lines and multiple PSPs coming in. The topmost one is an ePSP and the bottom two are iPSPs. So in this case the iPSPs dominate, and therefore they inhibit the neuron, it does not get excited, and there are no action potentials that are released. Whereas in the second schematic you can see that the topmost line is carrying two ePSP waves, and the middle line is carrying one ePSP wave, and the bottommost one is carrying like a single iPSP wave. So in this case the ePSPs dominate. So all these waves, when they arrive at the soma, okay, they tend to excite the neuron, and it produces action potentials which propagate along the axon. So this is in summary how a neuron functions and how a neuron generates signals. So in summary we can say that brain is a complex and dynamic network. It is not only very large network, the network is very complicated, is very labile, it is varying constantly, and also the neurons produce signals because neurons are electrochemical signals. They can generate and send signals both electrically and chemically, and there are 4 components of neural signaling. 3 of the components which occur within the neuron's body, right, are electrical signaling components, whereas the signal that jumps from one neuron to another is a chemical signaling step, and neurons exhibit this all or none response in the form of action potentials, that is either respond or not respond. There is nothing, it is not a graded potential, it is not a graded response, whereas we have seen that the PSPs, both ePSPs and iPSPs are graded response, and neurons perform a temporal and spatial summation of the inputs received from the other neurons. So in this segment of the lecture we have looked at a kind of a very simplified outline of what kind of signaling occurs in a neuron, right, but we did not discuss how these signals are generated, what is the molecular and cellular machinery that generates these kinds of signals, because I thought it makes it easy if you just look at the outline first and then go deeper into the various mechanisms and see how these signals are generated, because neural signaling is a very big subject, so quite a complex subject. If it straight away go into the molecular mechanisms, it looks all complicated, it's the chance of getting lost. So we thought that it is easier to look at the outline first and then go deeper into it in the next segment of the lecture. Thank you.